Thanks and welcome to the Biogeochemistry Science Friday. Our first speaker today is Chen from Lawrence Berkeley National Laboratory. Take it away, Chi.
Okay, yeah. Thanks everyone for coming. It's my pleasure to share my recent research to you. My name is Chen, and I'm a postdoc at the Berkeley lab funded by the Rubisco project. So this Wednesday, you may have saw Trevor's paper published in Nature, which is about identifying a historical emergent constraint on CO2 fertilization effect on global photosynthesis. Today, I'm going to talk about diagnosing the direct CO2 fertilization effect from FluxNet site and using remote sensing observations. I would first like to thank my collaborators who provide me with tremendous support in this research, Berkeley, Colin Prentice, and Trevor Keenan. So the central question of our research is the CO2 fertilization effect, or CFE. We now have globally distributed networks of long-term eddy covariance measurements, which could contain the information of CFE. The question is, when and how can we detect a signal of CFE emerge in these measurements? The second question is, can we accurately constrain the CFE using satellite observations and global meteorological reanalysis data? Next, let me define the CO2 fertilization effect. So I'm going to talk about the CO2 fertilization effect on terrestrial photosynthesis. Terrestrial photosynthesis is quantified by the terrestrial gross primary productivity, or GPP. The process of photosynthesis includes the exchange of CO2 and water flux between the land and the atmosphere. Both fluxes can be described by the Ficking gas diffusion. For example, I'm showing the equation of CO2 flux during the photosynthesis. Fc is the CO2 flux, G is the stomata conductance, Ca is the atmosphere CO2 mole fraction, and Ci is the leaf intercellular CO2 mole fraction. According to the CO2 measurement from NOAA, the atmosphere CO2 is increasing at a pace of 2.1 ppm per year in this century. According to the displayed equation, an increase of the atmosphere CO2 will lead to an increase of the carbon flux GPP. We call it the fertilization effect. While the CO2 fertilization effect on GPP is more complicated than the first order estimate from the equation I just showed. During the photosynthesis, there's a water loss along with a carbon gain. Plants, they are smart and they actively adjust stomata conductance G and leaf intercellular CO2 concentration, Ci, to optimize the gas exchange problem. We call this phenomenon optimization. So plants are doing a carbon water economy, maybe more complex than that. For example, they are also thinking about the nutrient economy, which will be mentioned shortly in the later slides. Estimating the direct impact of CFE on terrestrial GPP is challenging because all GPP are modeled, not directly observed. This is because a fraction of carbon gain will be consumed immediately by ecosystem respirations. And the internal variations or trends in GPP are subject to confounding effects of climate variability and model representations. The Earth system is so complex, many things are covariant at the same time. Thus, when we are studying the CFE effect, we should be very careful to make sure that we are looking at the actual CFE effect. This figure on the left gives you a sense of how different the GPP magnitudes are for different GPP products or models. And this figure on the right shows how different the GPP trends are among different trendy dynamic global vegetation models. So a promising way to exclude the confounding effects of the climate variability is to constrain the GPP analytically. Then we can take the partial differential sensitivity of GPP to the CO2. By this way, the CFE effect is strictly defined mathematically. And in this study, we are going to use the eco-evolutionary optimality theory to constrain the GPP. Here, the partial differential sensitivity is denoted as beta CO2. As there are several unknowns in the gas exchange problem, for example, the carbon flux, stomatic conductance G, and the leaf interior CO2 concentration, we need to introduce these optimization theories to constrain the problem. And each theory can provide one additional equation. And these theories are widely tested and supported by many observations or experiments. In our approach, first we use the Cohen-Farquhar hypothesis to constrain the stomatic conductance G. Second, we use the least cost hypothesis to constrain the intercellular leaf CO2 concentration CI. We then equate the equation of either G or CI from the solutions of these two hypotheses. So we can constrain the marginal water use efficiency lambda, which is a previously unconstrained but important parameter in the optimization processes. And note that we also need the Farquhar photosynthesis model to constrain these variables. Keep in mind that the problem can only be solved once the number of equations you can write is equal to the number of unknowns you have. The next question is to constrain the photosynthetic capacity. So this is the Farquhar photosynthesis model. Vc max and J are the photosynthetic capacity. Vc max is the maximum rate of Rubisco activity, and J is the rate of electron transport. The Farquhar model separates the photosynthesis into two regimes, light saturated and the light limited. And in operation, the actual photosynthesis should be the smaller of the two. To constrain the reference Vc max and the J max, we need to consider the impact of plants that balance the nutrient allocations. So we apply the coordination hypothesis to the averaging environment of the peak LI months during the study period. So the climatological mean carbon assimilation rate AC is equal to the counterpart AJ. With such implementation of the coordination hypothesis, we do not need the information of biome types. And our GPP can still remain either light saturated or light limited at a short time scale. The last step is to deal with the canopy upscaling problem. So we calibrate a big leaf canopy upscaling factor for each FluxNet site. We use the least square method to find the best fit for the canopy upscaling factor for each month. After calibration, we set this canopy upscaling factor as a constant with no interannual variations. So this guarantees that our calibration will not confound the diagnosed CO2-induced GPP trend. So the trends are not introduced by this calibration. Finally, the GPP and the CFE are analytically constrained by 7 measurable variables, including the atmosphere CO2, satellite-derived leaf area index, air temperature, soil water content, specific humidity, incoming short wave radiation, and surface pressure. One novel point is that we also introduced a modified logistic function of the soil water content to the marginal water use efficiency, which allows the soil water content to directly participate in the optimization of the carbon water nutrient economy. Due to time limitation, I cannot talk about the details here. But by the way, I want to mention that in this study, we used the canopy upscaling factor to make our model to be parsimonious, and it is easier to take the partial differential sensitivity of GPP to CO2 and to other factors. We actually have a follow-up study which employs a full canopy radiative transfer and solve the energy balance, but the same EEO model. That study proves that our EEO framework itself can accurately capture the seasonality in photosynthesis, which does not require any parameter calibration. So let's look at our results at the FluxNet sites. This figure shows the averaged GPP anomaly. So to get the average, to get the overall average GPP anomaly, we first calculate the annual summed GPP for each site. Then we calculate the site level annual anomaly. After that, we average the anomaly across sites. Then we test the overall GPP trend. The results show that our EO model can well reproduce the overall GPP trend and the inter-annual variability in the eddy covariance measurements. The EO inferred GPP is showing in red, and the EC eddy covariance inferred GPP is showing in black. The overall GPP trend is around 8.3 to 9.1 gram carbon per meter square per year square. The reason why I have a unit of the per year square is because the flux GPP is a flux term. It should be mass per meter square per time. And now we are looking at the trend, so the time should be the year squared. A unique aspect of our EO model is that it allows us to attribute the GPP trend to different factors. For example, this figure shows the results of a univariate GPP trend analysis. Univariate means that we only allow one variable to change, but keep other variables as their climatological means. The first three violins in gray show the total GPP trend. From left to right, they are eddy covariance inferred, our EO model inferred, and the aggregated GPP trend for each individual variables from our univariate sensitivity analysis. These three violins are expected to be consistent. The distribution, the probability distribution in the violin is generated by bootstrap resampling, which is to test the influence of missing data to the trend estimate. We randomly resample the data many times. For those colorful violins, they are the trend attribution to different variables. You can see a dominant contribution from CO2 to the overall upward GPP trend. The estimate for an absolute CFE effect is about 4.5 gram carbon per meter per year square. You can also see that soil water content, specific humidity, and temperature can make an important contribution to the total GPP trend. But the contribution from leaf area index and incoming shortwave radiation is small. As I mentioned earlier, our model is able to diagnose the CFE for each site using the partial differential approach. Unlike the univariate analysis, which requires a long time to ensure the GPP trends to be statistically significant, the analytical approach allows the estimation of CFE even for a short observation period. And naturally, this entangles the confounding effects of climate variability. So you can see that we diagnose a median CO2-induced GPP trend at about 4.9 gram carbon per meter square per year square. And this value is very close to the value diagnosed from the univariate sensitivity analysis I just mentioned earlier. And in this plot, each red point represents a site. Similarly, our EO model allows us to attribute the GPP inter-annual variability to different factors. That's why we know that this is detrended. And the ability of each factor to cause the GPP inter-annual variability is defined as the partial differential sensitivity of GPP to that factor of interest times the inter-annual standard deviation of that factor. And in our study, we find that soil water content is the largest contributor to the GPP IAV, inter-annual variability, followed by specific humidity and temperature. But the contribution of CO2 on the GPP IAV is relatively small. Finally, we upscale the EO model from FluxNet sites to the global scale. This figure shows the diagnosed beta at the annual scale, from which we can quantify the magnitude of CFE across biome types and growing climate. The inputs of this map are ERA 5 meteorological forcing and MODIS area. We also use multiple satellite GPP products to calibrate the canopy upscaling. The global average beta is around 4.4 gram carbon per meter square per year per ppm CO2. You can see that the largest CFE is expected in the evergreen tropical forests in Amazon, Congo, and Indonesia. Let's look at the within biome beta variation, which is impacted by both climate and CO2. But it is worth to note that CO2 in our EO model is prescribed without spatial variations. Therefore, the spatial variation in beta CO2 is solely driven by the climate. These bars represent the mean beta value for each biome types. The spatial variation of beta is shown by the black arrow bars, which is one standard deviation of the beta CO2 within each biome type. This is due to different background climate, even in the same biome type. The largest spatial variation is seen in the biome type SW, which consists of shrubs and savannas. These plants are widely distributed from the circumpolar to pan-tropical regions with the greatest climate contrast and therefore the largest spatial variation in beta CO2. An interesting finding is that the diagnosed beta CO2 do not have a significant interannual variability, which is shown by the red arrow bars. That said, the recent increase in CO2 or the temporal fluctuation in climate should not lead to a large change in the CO2 fertilization effect. This contradicts to the finding of a recent paper published in Science, which estimate a 40% drop in the CFE based on their regression analysis. Then let's look at the magnitude of the relative CFE in our analysis. The relative CFE is defined by the trend in GPP caused by CO2 divided by the background GPP. And this table shows the magnitude of relative CFE with different calibration data and in different biome types. Let's look at the first row of the table. We can see that if we use the ensemble mean of 8 satellite-derived GPP products to calibrate our canopy upscaling factor, then we would expect a relative CFE at about 4.1% per decade for all biome types. As I mentioned in the flux site analysis, the calibration itself does not introduce GPP trend, as the calibrated parameter has no year-to-year variations. But the calibration can change the trend magnitude. An important issue is whether our global estimate of CFE is robust. To address this question, we use different satellite GPP products to calibrate the canopy upscaling factor of our model. In the relative fashion, we can see that the magnitude of CFE is actually quite conserved, regardless of the data used to calibrate our model. And they are quite similar in all C3 vegetations, including the evergreen barley, other forests, short woody vegetation, grasslands, croplands. Last, we compare our diagnosed GPP trend and the CO2-induced GPP trend to dynamic global vegetation model simulations and to satellite-derived GPP products. This figure shows relative GPP trends, which is the GPP trend divided by the background GPP. We have 5 bars. Bars in gray show the total GPP trend, which is caused by the combined effect of climate and CO2. Bars in pink represent the CFE, which is the CO2-induced GPP trend. We also separate the bars into 3 groups, A, B, and C. Group A are the trends diagnosed by our EO framework. Group B are the trends diagnosed by trendy V6 DGVMs. Group C is the trend in satellite GPP. Satellite GPP only produce the total GPP trend. So we also scatter the individual models or satellite GPP products in color-coded points. For our diagnose, the CFE, our CFE is at the upper end of the trend distribution of their counterparts. For DGVMs, we can see that like Cable, DLEM, Julius, and also ISAM have relatively strong CFE, the B2 pink bar. In satellite-derived GPP products, we can see that those products based on terrestrial biosphere model like BESS, and P model show relatively strong GPP trends. An exception is for the model C6. They also show strong GPP trends. The greatest disagreement between our EEO framework and other models comes from the tropical evergreen broadleaf forest. Our study assumes that evergreen broadleaf forests follow the same eco-evolutionary strategies in optimizing their carbon water nutrient economy as that in the other C3 vegetation. We limit the total nutrient availability implicitly in the leaf area index. As I mentioned earlier, our EEO model diagnosed a similar relative GPP trend caused by CO2 across C3 biome types. This is shown by this horizontal dashed line in blue. Among the DGVMs for the evergreen broadleaf forest, we can see that like cable, LPJ gas, and perhaps ISAM, they also show a strong CFE in the tropical forest. But for the satellite GPP products, they show much weaker GPP trends in the evergreen broadleaf forest because their GPP trends are depend more or less on the satellite reflectance. But the reflectance of these tropical forests is almost saturated, which makes their actual GPP trends kind of elusive. Take-home message. So using optimization theories, we provide robust observationally inferred evidence that a strong CO2 fertilization effect is detectable in globally distributed eddy covariance networks. This CO2 fertilization effect can also be constrained at the global scale using remote sensing data. Our framework further provides the opportunities to decouple the confounding effects and diagnose the sensitivity of GPP to multiple factors other than CO2. So I will stop here. Thanks for your listening. I'm happy to take questions.